In this video, I'm going to show you the effects of distilled water on red blood cells under a microscope. I've placed a drop of distilled water on a glass slide along with a normal saline solution on another to show what blood cells should look like normally. Normal saline is mostly sterile water with a concentration of 0.9% sodium chloride, or more commonly known as salt. I'll be using a lancet to obtain a small amount of blood from my finger, which is just a tiny spring-loaded needle that only breaks the first layer of skin. Now that we have our samples, we can take a look at the difference under the microscope. I've added the magnification and which sample I'm using at the top right of the screen for reference. Looking at the saline sample first, you can see that red blood cells normally look like these small biconcave discs that are packed with hemoglobin, which is a protein responsible for gas exchange, giving blood its red color, and many other functions. There are some deformed red blood cells, but for the most part they look normal and haven't lysed or lost any of their hemoglobin. Switching over to the slide with distilled water, you can see that most of the cells have this swollen, disfigured appearance, and this is due to distilled water being hypertonic, which causes water to rush into cells and eventually burst from a process called hemolysis. When the cells burst or lyse, they also release all of their hemoglobin into the sample, which is why there is a slight tint of orange throughout the screen. You may also notice outlines of circular cells that are barely visible all over the slide, which are known as ghost cells, the empty husk of red blood cells that have lost most, if not all, of their hemoglobin. Even though the cell contents are empty now, it still maintains a similar shape due to structural proteins like spectrin and actin, which form a cytoskeletal mesh that provides support to the membrane. Think of it as a high-tech tent with bendable and resilient poles that represent the network of proteins. Since the poles are bendable, it allows the tent or cell membrane to temporarily deform, then return back to its original shape as needed. It's not an exact match, but hopefully it gives you a sense of how blood cells maintain their structure while maneuvering through the body. Going back over to the slide with saline, we can see that the cells still look similar to how they did when we initially looked at them. The reason why red blood cells stay alive slightly longer inside the saline solution versus the distilled water is because the saline is isotonic, meaning it has a very similar osmotic pressure to that of our blood, which prevents excessive water movement into or out of our cells. Just to show a different visual of how cells act in distilled water versus the normal saline, I put a drop of blood in a vial of distilled water and a vial of the normal saline solution, then time-lapsed it. This is around 3 hours of footage scrunched into a 15-second clip, but you could see a difference almost instantly in real time. The vial of distilled water has a lot more of a uniform red color, while the saline solution's color separated. This is because the blood cells in the vial of distilled water have all lysed and released the hemoglobin into the water, coloring it red, while the cells in the saline stayed preserved and simply fell to the bottom. Even a little over 3 hours later, the cells I took from the saline solution still retained that classic biconcave shape for the most part and still contained hemoglobin. In the sample of water, the ghost cells here are a lot harder to see without a lighting method like phase contrast, since they have all fully lysed and are completely transparent. I also added some distilled water to some slides with saline to try and show in real time what they look like lysing, but was only able to capture it at lower magnifications due to how chaotic everything is when adding the water and how fast the lysing occurs. This clip is at 200 times magnification and phase contrast, and you can see that some of the blood cells turn a dark color, then eventually transparent as they lose the rest of their hemoglobin. It was almost too difficult to see in bright field, and the conditions had to be just right to see the ghost cells. To finish this video up, I've also made a hypertonic solution and added it to the red blood cells to show the effects of the different osmotic environments. In a hypertonic solution, there's a higher solute concentration, or in this case salt, which results in the water moving out of the cells and shrinking the membrane. It's essentially the opposite of hypotonic. All I did was add a little bit more salt to one of the isotonic saline solutions. These cells are known as crenated or Burr cells, which are red blood cells with shrunken membranes that have lost their water. The membrane, while flexible, still has limits and can't shrink uniformly, so it crumbles and folds over itself, creating those spike projections you see. So now we've seen how red blood cells react in an isotonic solution, a hypotonic solution, and now a hypertonic solution. Understanding these reactions is crucial for medical practices like administering IV fluids, so doctors can decide whether to hydrate cells or balance fluids and electrolytes in the body. I don't have any kind of merchandise for sale, so subscribing is the best way to support the channel at the moment. I'll play some extra footage for the rest of the video with the labels at the top right corner, and as always, thanks for watching.